Hello, in this video, we are going to be talking about microbial diseases. So, as the name suggests, we're going to be talking about diseases that are caused by microbes, but we're not actually going to be talking about specific microbial diseases. That will be something that we cover later in another video. This is just kind of going over a brief overview of how microbes actually cause infections. So, first of all, how do we prove that a specific microbe causes a disease? I know it seems pretty intuitive nowadays, but back in the day, we didn't actually know microbes existed. So, when we first discovered microbes, we had to prove that, hey, this bacteria causes this disease, and then this bacteria causes that disease. And so we had to prove that. And there was a person named Koch who created these postulates that essentially prove whether or not a specific microbe causes a specific disease. So the first rule of Koch's postulate is that if you have a healthy organism, that organism, for example, a healthy mouse, should have no microbes inside of their blood or whatever it is, whatever sample we're collecting. But if we have a diseased organism, an organism that is sick, it will have the suspected microbe, if we look under the microscope. Now, this is the second, I guess, requirement, is that we should be able to grow that microbe in the lab from a sample, and we should be able to isolate it. And then that isolated bacteria, we should be able to inject into another mouse. So here's a different mouse that's healthy. And that mouse should acquire or kind of have the same symptoms as the first mouse. So whatever kind of disease, whether it's, a fever, coughing, or whatever, that mouse should have the same illness. And then it seems kind of redundant, but this is the fourth requirement, is that from this mouse that you have infected, you should also be able to isolate the microbe, and it should be the same microbe as from step two. And so those are the postulates of how we can essentially prove that a specific microbe causes a specific disease. Now, we obviously know that there are many microbes that are hard to grow in the laboratory. So, for those microbes, this doesn't work. And we also know that there are some microbes that exist in a person which don't cause disease. And so, obviously, that kind of breaks some of the rules. But for the most part, generally speaking, and 90% of the time, Koch's postulates is used to prove that a specific microbe is what causes a specific disease. So when a individual gets infected with a microbe, we're going to go through these stages of disease. So incubation is just the first stage. Your pathogen, whether it is a bacteria or a virus or whatever, is going to enter the body, and then it's going to multiply. Now, in this graph, the red line is showing the amount of the pathogen, so whether it's a bacteria, a virus, etc. And then the blue line is showing the symptoms that an individual would be experiencing. Now, notice that during incubation, there is a small increase in the number of the bacteria, virus, etc., but there is no symptoms yet. So that is the main kind of key factor in incubation, is that we don't have any symptoms. Then we go into the prodromal stage, and this is where the pathogen starts to multiply more, and you start to get some of your early signs and symptoms, whether it's fever, scratchy throat. Again, it depends on the microbe and the disease, but you would have some kind of symptoms. Then you get your illness, and this is where your symptoms are the most severe and the microbe is at its maximum number inside of the person. And then, of course, the individual starts to get better. Their immune system starts to fight the microbe. And so we have this decline. So the number of pathogens declines, the number of symptoms declines. And then finally, we get to convalescence, where the body returns to normal. And so at one point, you're not going to have any more symptoms. And then the amount of microbe is, low enough. So those are the 5 stages of disease. Now, how do microbes even enter the human body? Well, there's different ways, and we call these the points of entry, right? This is how a microbe infects us. They have to get inside. And so a common one are your mucosal surfaces. So any surface of your body that is exposed to the outside that has a mucous membrane. So for example, the inside of your mouth, nose, eyes, ears, that's mucous membrane. So if you eat something, or if you inhale something, or you touch your eye, etc. You can get microbes in those surfaces. But then there are other ways of getting in, and so we call these the parenteral route. Basically, it is breaking the skin or mucous membrane in a different way. So, for example, if you have a cut, then they can enter through broken skin or a needle, or an insect bite where it's more forceful. It's not just the microbe entering your mucous membrane naturally, but there's like some kind of force, whether it's a cut, a needle, an insect bite, etc. And then, of course, there are other ways that microbes enter through the genital regions. So these are going to be your sexually transmitted microbes. That is another way that they can enter. Now, when we are describing a specific microbe, we describe it based on its virulence. And, there's a couple vocabulary words that I want to get out first. So pathogenicity is just the ability for a microbe to cause disease. Some microbes are pathogenic, some microbes are not. And virulence is how much it is pathogenic. So the level of pathogenicity. Is it really infectious or is it not, essentially? And there is a way for us to quote unquote measure how virulent a microbe is, and we call it the LD50. So the LD50 stands for the median lethal dose. It is the number of microbes that you need to be exposed to that would cause death 50% of the time. So median meaning the middle. So for example, if this is a specific microbe and they did a test and they exposed, again, let's just say they did it with mice. They exposed mice to, very little amounts and very high amounts of the bacteria or virus. Of course, the mice will get sick. So all of the different mice will get sick. But the mice that were exposed to a small amount of the virus or bacteria, they probably survive. And so, again, this is the percent mortality. So, how much of them were dead and how much of them did not die. And so, basically, the LD50 is the point at which about half of the mice will die when you expose them to that number of pathogen, whether it's a bacteria or virus. And so, this kind of gives us a metric of how pathogenic a microbe is. There are some bacteria, for example, where if you are exposed to 5 cells, so 5 bacterium, you will most likely die. Whereas some microbes, you have to be exposed to millions and millions of them before you actually die from that disease. And so that is kind of this is our metric of how virulent a microbe is. I hope this video was helpful, and I will see you in the next video.